Hello students, today we will understand other three organelles present in eukaryotic cells. Those are ribosomes, microbodies and cytoskeleton. Ribosomes are small subspherical granular organelles that are not enclosed by any membrane. That means these organelles are non-membrane bound structures. These organelles are also present in prokaryotic cells. Ribosomes occur in large number in a cell. A cell may contain up to 500,000 of ribosomes as per their activities that they will be performing. Ribosomes are synthesized in the nucleolus, that is the part of the organelle nucleus, and from here the ribosomes are transported in the cytoplasm. Ribosomes are either found free in the cytoplasm or they are bound to an organelle, that is endoplasmic reticulum. Each ribosome have two subunits, larger subunit and smaller subunit, and these subunits are scattered in the cytoplasm. Depending upon the size of sedimentation coefficient, the ribosomes are of two types: prokaryotic ribosome 70S and eukaryotic ribosome 80S. We should remember S stands for Svedberg units, that is the measurement of sedimentation rate during centrifugation, and this we had learnt in detail in the prokaryotic ribosome which we had studied in the previous video. The 80S of eukaryotic ribosome have larger subunit of 60 Svedberg units and the smaller subunit as 40 Svedberg units, and together the sedimentation coefficient is 80S. Ribosomes also consist of highly folded rRNA molecules, that is ribosomal RNA molecules, many proteins as well as magnesium ions that play very important role in the protein synthesis. As we had discussed earlier, ribosomes are free or attached. The larger subunit is 60S, smaller subunit is 40S. These subunits are scattered in the cytoplasm, but at the time of protein synthesis, both the subunits join together to form ribosomes on the mRNA. We can call ribosomes as the protein factories as it is very essential for the synthesis of various types of proteins. It also provides the site for the attachment of tRNA and mRNA which participate in the protein synthesis. The next organelle are the microbodies, which are minute membrane bound vesicles like structures present in these cells. These microbodies utilize the molecular oxygen and helps in the oxidation process. Microbodies are present in both plant as well as animal cells. Microbodies are of two types, peroxisomes and glyoxysomes. Peroxisomes are also called as oxidative organelles. Peroxisome contain peroxide producing enzymes, that is oxidases, and also peroxide destroying enzymes, catalases. Oxidases enzyme catalyze the oxidation of biomolecules such as amino acids and uric acid producing hydrogen peroxide, and hydrogen peroxide is broken down into water and oxygen by catalases enzyme. Hydrogen peroxide is very harmful in the living cell. Therefore, it is very essential to break down this hydrogen peroxide into water and oxygen. Therefore, peroxisome play very important role in these cells. It also produces cholesterol and phospholipids that are found in brain and heart tissues. It helps in lipid metabolism too. In plants, it helps in recycling of carbon during photorespiration. Thus, plant peroxisomes are found in photosynthetic cells associated with chloroplast. Glyoxysomes are microbodies which contain enzymes that can convert the fatty acids into carbohydrates. They are quite common in the cells of germinating oilseeds such as castor, watermelon, peanuts, etc. and some fungi. The enzymes that are present in the matrix of glyoxysomes helps in the conversion of the fatty acids or beta oxidation of fatty acids to produce acetyl coenzyme that gets converted into carbohydrates. As our skeletal system works as support structure for our body, gives the body a proper shape, likewise the cells too have their own supportive framework inside it, and that is cytoskeleton. That means it is the skeleton of the cell. It is the elaborate network of filamentous structures in the cytoplasm made up of proteins. These cytoplasmic structures that are made up of protein elements are found in eukaryotic cells and some in blue-green algae. Cytoskeletal structure are of three types: microfilaments, intermediate filaments and the microtubules. The microfilaments are the ultramicroscopic, long, thin, cylindrical rod-shaped proteinaceous filaments. These microfilaments are the part of cytoskeleton to support the fluid matrix. It also brings about the muscle contraction. Intermediate filaments are the supportive elements in the cytoplasm of many animal cells. They have non-motile structural proteins that maintains the shape of the cells and keep the nucleus and other organelles in place. Microtubules are elongated, unbranched cylindrical tubules. These tubules can be single or in bundles in the cytoplasm. They help in maintenance of the shape of the cell. In general, cytoskeleton gives the mechanical support, helps in motility and maintains the shape of the cell. They are the major constituents of spindle fibers, chromosomal fibers, centrioles, basal body of cilia and flagella. So this is all about the three organelles that we have gone through today.